we come to the interesting part of this talk. And that is the hypothesis. And it comes a little bit to the question we had before. What do I do with exploratory secondary, right? So there is exploratory secondary is one thing, but we're going to talk about exploratory secondary on Wednesday. Today I'm going to talk about the primary hypothesis and the primary hypothesis of your research question. And research questions in clinical trials is only one. It is always hypothesis driven. It's not, I want to estimate what is the effect of this and that. No. Or is there an association between the, no. You're making a claim. This is better than that. This is equivalent to that. This is not worse than that. And, or we talk about bioequivalence. So you are making a claim. So unlike other types of research studies, the observational studies, where you may have an exploratory research question, here it's always a testing of a hypothesis research question. And the research question then is going to be answered by a test of a hypothesis. And all of you, I assume, have taken some basic statistics course, and you know about hypothesis testing, and you know about statistical significance, and I assume everybody knows and adores the p-value. Well, I will hopefully try to teach you that the p-value is not the answer in a clinical trial. It's one of the little things that comes out at the end, but it is not the essential thing. The essential thing is understanding what is your hypothesis. So most of the time, we do what are called superiority trials. We're trying to show that A is better than B, or this new thing is better than what is the usual care. It used to be that we could do some intervention versus placebo, new drug versus placebo. Most of the times nowadays, it's almost unethical to do that. When a new thing comes out, a new disease comes, and we don't know what works, then and only then it is okay to test against placebo. So for example, when HIV appeared in 1981, 1982, there was absolutely nothing that worked. And they're developing drugs. AZT was one of the first drugs. And it was okay to do AZT versus placebo because nobody had anything that worked. But once they showed that AZT worked, drug B comes along. Drug B cannot be tested against placebo because we already have something that works. It may not work perfectly, but it works. So therefore, now the new drug has to be tested against the AZT, the usual care at that time. So we'll talk about why superiority trials are preferred. And sometimes then we'll see why they are not so preferred nowadays. So the goal here is to show that the effect of one treatment on some endpoint, some clinical event or clinical endpoint, change in weight, could be death, could be cancer remission, can be learning a new technique, how one treatment is superior to that of another treatment. So we do that in placebo-controlled trials, trials to show an active is superior to another active therapy, like it lowers cholesterol more, it does this better, it does that better, etc. So the hypothesis for superiority, and I unfortunately will use some Greek letters. We use Greek letters for only one reason. That is to confuse people. So therefore, you hire us and we have jobs as statisticians. That's the only reason. We could use other letters, but then you won't hire us. Okay? So we have the hypothesis for superiority is that we believe that the treatment groups are, at this point, are equal. It's the status quo. There is no reason to believe that the new treatment is better than the current treatment, or that the difference in whatever effect is the same. So the difference is zero. And we're interested in showing that they are not equal. Now here comes a question. You say, wait a second. If you want to show superiority, why is it that are you doing it two-sided? And because two-sided means that if I was doing this in the bell-shaped distributions, of normal distributions, then what we're saying here is, let's say that currently we're over here, and this is the difference. So we're saying the difference between the distributions is centered at zero. And now I'm going to reject if I get some difference that's way up here, or if I get something that is way down here. And you say, but wait a second, you said superiority. This would be that it's worse. I don't really want to show that. I want to show that it is on this side only. And I want to be certain, and we'll talk about hypothesis testing, we want to be over here with a very small probability. And usually we would want 0.05 here, because that's the magical number that we'll talk another day about why it's not really magical, but people believe it is. But what happens is that if I do it two-sided, then this is 0.025 here and 0.025 here. And this value, this is standard normal distribution, is 1.96, and this is minus 1.96. And I would reject my hypothesis if I get a Z value, so this is a Z statistic, that is on this side of 1.96, and say, oh, it's statistically significant. And you would not expect to get on this side whatsoever, because you didn't expect it to be worse, you expect it to be better. But what would happen is that many times you don't actually get 1.96. You may get, for example, 1.75. Let's say, I'm just making up that number. And if you calculate the p-value for that, it's going to be greater than 0.025. So it would be not statistically significant. But if you now say, oh, wait a second, I'm only interested in this side. So I was going to put here, and forget this. I'm not going to do this. That's stupid to do because I want to show it's better. The critical value here is 1.645. So now all of a sudden this is statistically significant. And what would happen was that back before the days where you had to register your protocol, people would say, we designed our study to be one-sided 0.05, and we got good result, but they actually had designed it two-sided, where it was going to go, and but they changed their mind. Nowadays, because people cheated in those days, we are all forced to do things that say you can do it one-sided. If you want to do it one-sided, oh, what happened? If you do it one-sided, you better do it at one-sided 0.05 and use that 1.96 and not do anything. Yeah, you have it open. Oh, I did it. Let me just get through. Sorry about that. So we do it two-sided, or if you want to do it one-sided, you do one-sided 0.025. 0.96 doesn't change, okay? All right, so if you're looking at means as your outcome, this is its t-test that you would do, or a z-test if you have a large enough observations. If you're doing proportions, you do a chi-squared, or you can statistic, and you can get the p-value by the t-test or z-test or Fisher's exact test. And very simple. And you say, wait a second. You said clinical trials. I came here to learn about clinical trials and the analysis of clinical trials. And it's very complex. And you just tell me right now that it's very simple. It's t-test or chi-square test. I did that in my first course in introductory statistics. I say, yes, it is very simple. The analysis of a clinical trial is the most boring, simple analysis possible. Why? Because you've controlled everything by design. You don't have to do fancy models. You don't have to do any of these things because there's no confounding. Only if I randomize the participants. I don't have to do anything fancy. This crude primary analysis of a clinical trial is as simple as that. If I've done a cluster randomized, then I have to adjust for the design effect. If I've done a stepped wedge, I have to adjust for the design. But I don't have to adjust for confounders unless I have residual confounding. That becomes a more complex thing. And the one interesting type of analysis is the interim analysis, and we'll talk about that on Wednesday. But in a superiority test, it's just as simple as that. If the p-value is less than 0.05, we reject the null hypothesis and say there's enough evidence to believe that these two groups are different. And so we say we have a significant effect. Maybe the battery's there. I think the battery's there. And if the p-value is greater than 0.05, then we say we do not have enough evidence to reject the null hypothesis. And we say the data we've observed is compatible with the null hypothesis. And I'm being very careful with my choice of words. We are not saying there is no difference, because saying there is no difference is equivalent to accepting the null hypothesis. And we never accept the null hypothesis. It's the same reason in every judicial court system in the world, maybe exception some countries, but at least in India, in Canada, and US, when somebody's accused of a crime, you are presumed innocent until proven guilty. If the evidence is enough to prove guilty, then the judge or the jury or whoever says, we have enough evidence, we reject innocence, you are guilty. But if they don't have enough evidence, the jury, the judge says, we find you not guilty. They don't prove your innocence. They say, we don't have enough evidence to say that you're guilty. You may be guilty. We don't know. But we never accept your innocence. We just say you're not guilty. Okay? Is everybody clear on that? The same analogy for the judicial system works with hypothesis testing. confidence intervals when testing for superiority. So what would we do? We would calculate our point estimate of the effect size and do a confidence interval. What kind of confidence interval? 95% two-sided confidence interval to be equivalent to the two-sided 0.05. So if we get a confidence interval that is on the other side of zero, does not include zero. What happened? Then we would say it's significant difference. The intervention effect is significantly better. If you actually were on the other side, it would be significantly worse, but you don't expect that to happen. And if the p-value is greater than 0.05, the 95% two-sided confidence interval will contain zero, right? They're one and the same. So there's a one-to-one correspondence between a 95% confidence interval, two-sided, with a two-sided test of hypothesis at the 0.05 level. That you all got, I hope, in your statistics courses. So, easy test for superiority. This is what we do probably 80%, 90% of all clinical trials are looking for superiority. But what happens is that sometimes you are not interested in the hypothesis of superiority. You just want to show that it is equivalent to something else. So, for example, you may be working in a pharma company. There's already a drug that's approved for something, and your company has developed a generic or has developed an alternative, and you want to say, our drug is just as good as the other one. It's equivalent to that. And buy ours, don't buy the other one because ours is cheaper or something like that. And or ours, you may cost more, but you only have to take it once a day instead of twice a day or whatever. So you want to show that it's similar. So to show similarity, we talk about equivalence trials, and it's to show that they differ not too much. That's what similar is, right? And so you want to show that there are two different formulations that are pharmacokinetically similar, or you want to do lot-to-lot consistency of a vaccine manufacturing process. So this comes through quality assurance kind of things. And so you may want to show also that the two interventions are similarly clinical. So what would you do? So we call this now bioequivalence because they want to show that they are bioequivalent to an already approved ICMR or FDA approved name brand drug. And so it'll have the same components, but it's manufactured by a different company. And it's what you do is you evaluate different drug concentration levels in the blood of healthy volunteers. Well, wait a second. I'm doing that. I'm not doing a trial. I don't have two groups. I'm not randomizing. I'm just giving this to some people, some volunteers, and seeing how the blood concentration goes up in the blood, how long it stays at the top, and then when it goes away. So why you call it a trial? I don't call it a trial. Some people call it bioequivalence trials. They are just bioequivalent experiments. They're not randomized controlled trials. So if the blood concentration levels are on average similar enough, and they have all sorts of metrics for it, then equivalence is assumed. And that's not what we would do as or consider as a trial. So bioequivalence, for example, they would look at how the blood gets taken in or how the concentration. So this is one thing that pharma does a lot. It's not a clinical controlled trial, but they have all sorts of parameters that they look at on this. So it's a, a valid study to do, but it's not a randomized controlled trial. So if we did clinical equivalence, what would that be? So it's not bioequivalent in looking at pharmacokinetic properties of the medication, but it's looking at clinical equivalence of that. Then we would have, again, the same parameters under one group and then the other. And then we would look at if there is a difference between them that is greater than some quantity delta. Again, another Greek symbol, hieros, right? To make you complicate. So if the difference between the parameters of the treatment and the reference are similar, hey, perfect. So if they are different by some quantity delta, then we will say that they are not similar, they're not equivalent. And so notice here that the null hypothesis is that they are different. Why? Because you want to show your claim is that they are not different. So when we are constructing our hypotheses, the null hypothesis is what we would believe at the moment without a study. And the alternative hypothesis is what I am claiming. So the claim is always there. So if I claim superiority, the alternative hypothesis is that it's better or that it's different. If I claim equivalence, then the alternative hypothesis is that they are not too different, that they are similar. The test and the reference are within very close to each other. And so the null is that they are different. And here we're doing that. So it's actually a sort of a reverse, right? And again, what is a test statistic? Simple. T-test, chi-squared, nothing strange. So what would equivalence trial be? Clinical equivalence would be that you've set this bounds delta and minus delta because they have to be, this is the absolute value, so it can go either way. And so equivalent would be that the confidence interval is within minus delta and plus delta. Okay, so not equivalent is if you go beyond that, or even if you go against one of those. Now, suppose that if you go this way, this is actually, it's better. The test gives you a better result, and here it would be a worse result. And now it's not worse, it is even better a little bit. So people don't like this anymore. People don't do this because what if I get superiority or it's a little bit better? Then I have to say it's not equivalent. So I don't do this equivalent trial. So almost never do you see anymore a clinical equivalence. All you see is bioequivalence if people are doing equivalence. So what you see are non-inferiority trials. So non-inferiority trials says, I don't want to show necessarily that my new medication or my new intervention is better than the one that is the usual care. All I want to show is that it is not worse. Why? Oh, so I'm offering you an alternative. Buy mine. Because it's not worse than that. Could be better, but it's not worse. And mine is cheaper. Mine makes me money, not them money. That's it. That's all I care about. Or it is easier to take. It's more available. It doesn't spoil so quickly, whatever reason. But the clinical effect is not inferior to whatever is there out there as a usual care. So how does this go? The show now that is no worse than an existing treatment by some amount. Which amount? So why do we use it? It's by showing non-inferiority compared to a treatment that is known to be efficacious, or having another advantage such as less frequent dosing or better tolerated than others, or cheaper, for example. So why do people do this? Not showing superiority? Well, maybe because you have less adverse events. It's ease of administration, it's less costly, but also, very importantly, this is used a lot in cancer research. It's just to say, we have another alternative here that you could try, because maybe when they're doing chemotherapy, this is not working for this patient. Well, here's another thing you could try. Here's another one you can try. Here's another one you can try. So it's good for the patients to do these sorts of things. So the other way these non-inferiority trials are sold to investigators, oh, you can also test for superiority. Well, that's what I would call an advertisement by a used car salesman. Don't believe it. It's not always true what they say. So you have to be really lucky and have something that is really superior. And you can have different types of parameters. And what you do is you do a one-sided confidence interval. But again, 97.5, so it's equivalent to the two-sided 95%, because you're only concerned with limits on the inferior side. So let's see what this looks like. So we have now, so far, 3 different types of hypothesis. The traditional, easy one that everybody knows, the superiority. We have the equivalence. Now I've changed that delta for gamma to confuse you more, because I've kept the delta for this one in the non-inferiority one. So what is this like? Let's go slowly through this. For superiority, I want to show that the intervention is different from the control by an amount that is the at least meaningful difference. We call that little delta. And this little delta is an important concept, because when we talk about sample size, when Jason talks about sample size, you will see that that is what drives the sample size. That's how effective do you think your treatment is going to be. And so, for example, let's take an example, and I will ask you, if you were said that some vaccine for COVID is effective in 90, let's put 87% in preventing serious, this is a standard vaccine is this. Then there's a new vaccine that comes that you want to claim is superior to that. Then I will ask you, how superior? Oh yeah, it's a lot. No, I need a proportion. What would you be happy with if it's on its effectiveness? How many of you would be happy if it's 100% effective? Everybody, I would assume. If it is 88% effective versus 87, do you think? That's not a meaningful difference. 1% is not meaningful. So I'll ask you, what if it is 90%? 87 to 90 is 3% difference. Is that meaningfully more? How many of you say, now you have to raise your hand. How many of you say that that is meaningfully more effective than that? 1 person, 2 people, 3, 4, 5, 6, 7. So about 7 people say that. So those of you that did not like 90, how about 95? Now you have 8% difference. Is that really much more? How many of you think it's that? All of you say that. Okay, so 95 is good. How about 93%? Now we're talking about 6% difference. How many of you think 6%? There's a few more, okay? How about if I went to 92%? Now I have 5% difference. Only one or two, three. So very few. So the smallest clinically meaningful difference here may be around 5%. This is the way to get that delta, by you yourself thinking what would make you happy if you see that. If you don't like to trust what makes you happy, ask your colleagues. What would make your colleagues happy? What would make the ICMR happy? What would make the FDA happy? What would make somebody happy? Don't ask the statistician to choose this, because it's not the statistician's job to decide what is a clinically important difference. Notice that there is no word significant here. There is no such thing as clinical significance, even though it appears in the literature. Significance is a statistical, technical word. Please do not use it unless you're talking about a statistical, technical definition. It's clinically important, clinically meaningful, clinically fantastic difference, clinically relevant difference, whatever. But it is clinical and it's subjective. It is your choice of what it is, okay? So that's the delta. Now, if I wanted to show something is equivalent, so now I have another different drug. And right now we have this standard vaccine is 87. And I want to say, I have another one that I want to show is equivalent. And this one is 87. And I say, oh, my new one, it is 82% effective. Is that equivalent? No, it's too much lower, okay? How about if I say 85%? It's 2% lower. Is that equivalent? Some people say yes, no. So you would want it to be 86% or maybe 88%. You don't want it to be 89% because that's more than 2. So the equivalence here is to be very similar. So now your delta, or you have a gamma, is 1%, while your delta here was 5%. To show something is equivalent, you want it to be very, very close. Okay? And that's, you'll see when we do the sample size, that this being small makes the equivalent studies very big, sample size wise. Okay? Again, people don't do that. They do the non-inferiority. And so I will say non-inferiority means I'm not going to be looking at that. These are the inferiority ones. And how much not inferior can it be? Will I tolerate, what I told you said 86, no, sorry. You said 85 is not, it's a little bit inferior. Is that too inferior or that's okay? 2%. Is it okay? So you're okay with 2%. Then if I ask you, wait a second, what about if this is 84%? Are you okay with 3%? You don't like it to be 3% less. It's your decision, not my decision. Is 3% less worse? Some people say no. Too worse. Too close. So maybe you're okay with 2%. So here, the delta, oops, was 2%. You're willing to tolerate being smaller than that. The gamma, you said it had to be 1%, and this delta was, you tolerated 5%. And you see that the non-inferiority studies tend to be larger in size than the superiority studies, but not as large as the equivalent studies. And that's what usually happens. There's this in people's mind, it is like that. And people need to be able to think this way what would make me happy if I found it? That's the way you choose these things to decide. So delta, capital delta, is the maximum tolerable difference for non-inferiority. You're willing to tolerate up to 2%, not more than 2%. Okay? Everybody clear on this? So this is a nice figure in this paper in General Internal Medicine that compares the different types of studies. So when you're looking for superiority, you want to have that your confidence interval is not touching 0, so that you are superior. Okay? And that's for a new therapy being superior. And if the, depending on what is your statistic that you're using, if the outcome is a bad outcome or the outcome is a good outcome, you'll have different types of which side you are, okay? So here, oops, sorry. If the outcome is a good outcome, so remission in cancer, curing somebody of the disease, then you want to be on this side. But if you're studying heart attacks or cancer relapse, or you're looking at something that's bad, like getting a stroke, then you want to be on this side. So the new therapy is better than that. And so because you're taking the difference between the treatment minus the reference one. And so you would establish superiority if your confidence interval doesn't include 0. Let's just stay with higher is better. So let's stay on this side. This is just the opposite. And if your confidence interval overlaps 0, superiority is not established. Now notice also the choice of words here. They don't say no difference. They don't say not significant and significant. This overemphasis on something being significant is the wrong way to understand these things. The proper way to understand these things is whether the possible range of values of the effect are consistent with it being superiority, or are consistent with it possibly not being superior. Okay? And equivalence, you have these very narrow ranges here, and this would have been gamma. Now we put a little delta here because I copied it from here. So that's another confusing aspect. But equivalence is only when you're within that narrow band. And equivalence, even if the new therapy is inferior, you're over here. But here, the new therapy is actually even superior, and you're on this side. Equivalence is not established. And the non-inferiority study, you would want that the lower bound of your confidence interval not go below here and below your minus delta. And here now you have non-inferiority. You have non-inferiority was not established. Here, non-inferiority was established. And the opposite if lower is better. Okay. So take a look at this. This is a great paper explaining it in clinical terms, how to understand these hypotheses. So why we do that? Because the goal is now to show that it's no worse than that of an existing standard. And so this is usually done for regulatory approval of a new investigational treatment, or show that an approved drug is no less efficacious than another, okay, or like a new dosing regimen and so forth. So these are commonly used. Why they're also commonly used with a new treatment that somebody comes up with is because of ethical reasons. As I said before, once somebody has shown there is a treatment that works for HIV, AZT is out there. Now, let's say you want to do triple antiretrovirals, you cannot do triple antiretroviral versus placebo. You have to do it against AZT. And if AZT is very effective, now you would have to show that this is even more effective than that. And the difference between, let's say, placebo has this effect and AZT has this effect, and I have my new drug, and maybe it's better, and I think it's going to be here. If I test it against placebo, I'll have a big difference. But AZT is here, and I have to test it against AZT. So now that difference is small. That makes my sample size go up. And that becomes very expensive. So now they want to say, well, I don't want to test superiority. I just want to show that it's not on this side of it. It's not too far this side. It may be on this side, but it's not too far on this side, closer to the other. Okay. So the question is, for superiority, we usually choose a delta from clinical judgment, but for non-inferiority, we can also do it by judgment like we all did here. But if you're going to submit a new drug to the US FDA, they have rules. And if you want to submit to them, you follow their rules. So I'll talk about their rules right now. So we talked this about already. So this is just another way of saying what we've just said. And this is a more detailed graph that if you have a good outcome, you focus on the lower bound and you don't want to have it go beyond this on this side. And this would be where you have it be inferior. You're compatible with inferiority. If your lower bound crosses the non-inferiority margin, but if it is the lower bound is above that, it doesn't cross the non-inferiority margin, you could say it's not inferior. And if you're lucky enough that this is actually all the way above zero, you can say you got superiority. But that you have to be lucky enough to get that. It's not that you can always get it. And you can do it for differences, or if you're looking at relative risks and you're doing ratios, you can do it also against 1 as opposed to against 0. If you like relative risk, for example, or hazard ratios or odds ratios. And the opposite for bad outcomes is on the other side. So take a look at these graphs when you can. So we've already mentioned that the margins are related, that logically it makes sense that the delta tends to be bigger, that the capital delta tends to be bigger than the gamma. So that this is 0, they are ordered like that. And way to think about it is, for superiority, that delta is the smallest clinically meaningful difference. For non-inferiority, that capital delta is the largest clinically tolerable difference to claim similarity. And careful, these terms you need to be careful of how you say these things. They have different meanings. So, common pitfalls. If you test for equivalence and you can conclude significant equivalent, this does not necessarily mean that if you had tested for superiority, you would not have found one, because you may have different deltas and gammas, for example. And if you test for superiority and do not find one, this does not mean that you can conclude equivalence, even if the power of the study to find the difference was high. So again, these things are not interchangeable, not compatible. They're different concepts. And how do you know which one you're doing? When you have your research question at the beginning. Remember what Tambo said? The research question guides everything. It guides which hypothesis you are testing. Okay? And remember, statistical significance has no relation with clinical importance of the result, nor its meaningfulness. It has absolutely nothing to do one with the other. This has to do with, can you rule out the role of chance? And the larger the sample size, everything is statistically significant. Any meaningless thing is statistically significant. All right. Now comes the big question that everybody asks me. Okay, I want to do a non-inferiority one. How do I select the margin, that capital delta? Do I do it like we did here? Yes, you can. There's no problem with that. But if you're working with pharma and you're going to submit to a regulatory agency, you better check in their guidelines what they want. ICMR may have some guidelines, FDA has some guidelines, the European Union has some guidelines. They may be similar. I haven't checked all of them, but you must check them if you're going to do them. But the first thing is you select them a priori before. This is part of your document that you register with the clinicaltrials.gov. And if you're going to submit to a regulatory agency, you check with them. This is how I'm going to do it. Will you approve my drug if we do this, if we show this? Talk to them. These people are going to be the ones making these decisions on your drug, and you don't want to have wasted thousands of rupees on this, crores of rupees on this. And it is a clinical decision. As I said, it's not the statistician who decides it. It's a clinical decision, but it has very important statistical implication, especially with samples. So often these were set by clinically relevant difference for each indication, and people would do it on their own. But then the regulatory agencies came out and said, well, if you're doing means, usually you have to say the standard expected standard mean or difference between standard and placebo. But most of the times people are looking at binary outcomes, difference in proportions or ratios in a multiplicative scale. And the US FDA had said that the margin should be for good outcomes 0.8 and for bad outcomes 1.25. That's what they originally said that they should do. Well, how do you select these margins? So then they had a revision in the current guidance for industry. This is, you can download this non-inferiority clinical trials establish effectiveness guidance for industry. This is not, unless you're submitting to FDA, this is not relevant, right? But this, if you're submitting to them, you better follow it. So they say you have to have two quantities. You find out what is the effect of the control. Does the control have some effect? This is the reference that you have. And this would be the effect that the control drug would be reliably expected to have between control and placebo. Where do you get that from? From the literature. And then you, or some previous study that somebody has done. Then you say M2 should be the largest effect of test compared to the reference that would be clinically acceptable. And it should be smaller than M1. Okay? The smallest difference which patients perceive as beneficial and which would mandate, in the absence of troublesome side effects and excessive cost, a change in treatment. So this is the minimal clinically important difference for them. They use that, clinically important. So that's M2, there's a clinical margin. And they have ways of finding that M2. And they have different methods of doing it. So they define M1, usually, let's say if it's a bad outcome, or the lower bound, so it's a good outcome, it's the lower bound of the 95% confidence interval compared in reference to placebo. And they say you set M2 as half of that lower bound to preserve 50% of the effect of the other one, of the usual care. And so they call it, if you want to preserve half of the effect, then you take half of that. Or you can be even more stringent and say, I want to preserve 75% of that. You have to choose one of those two. This will be even smaller. It will be a larger sample size. And that's difficult to do, right? And so in some situations, you may even, M2 may be smaller than that, but they will not accept anything that doesn't preserve at least half of the effect of the other. And then you could just do it. All you have to do is you find M1 and you just put that in a software. We'll talk about the softwares here and how to calculate it, and we'll calculate the sample size. And then you say, oh, my budget. That's where the question comes in. The budget happens. So here's an example where the minimum, the large value is good. And so this is we looked up the placebo, reference to placebo from a meta-analysis at this confidence interval. So minus 4.5 is the minimum of the reference effect. And we wanted to preserve at least half. You would use minus 2.25. Or if you want to preserve 75, it's minus 1.125. And now you just calculate what you would want as the, so this was this is 0 over here. And so you'd want to get either this, you calculate the sample size required for this difference or for this difference here. And it becomes whatever it turns out to be. And so we can do that in the multiplicative scale. And now I've changed it to large values are bad. And so the multiplicative scale, let's say the confidence interval of the reference versus placebo, relative risk goes from 0.7 to 0.8. So the reference is a good thing. And you convert this to risk reduction because it's easier to work on risk reduction. So it's 30% and 20%. So 20% is the risk reduction, the minimum effect of the reference versus placebo. So now upper bound of that is, that's why they set it at 0.8. So that becomes 1.25. That's the minimum of the reference effect. And if you want to preserve at least half of that, now you're going to take 50% of that. You better use a margin of 1.11. And the math is very simple to do this. Right? But the concept of understanding, I want to preserve some of the effect of the reference drug or reference intervention versus placebo. And again, you can go through this. That guideline will give you all the more details than what I've put over here. This is actually a study that we did recently. It's thanks to this physician who gave me this. He had to determine the non-inferiority margin of test treatment was factor 11 plus aspirin, and the active control was rivaroxaban plus aspirin. He was the principal investigator that showed that rivaroxaban plus aspirin was beneficial. So that became the new control. So he had to do that. And so he had to preserve that effect. And he had the effect was M1 was this. And so one way of doing it is you can take a bunch of studies. He included his study and got from that, gets the meta-analysis, it gets this, that's the M1. And now he wanted to preserve 75. So now M2 becomes very small. And then you calculate based on that. Okay? So these things are done in this non-inferiority states. So he got, we get the M1 there, and to preserve 50%, he would take half of it. It was reasonable, but he then changed it to event rates, and he had to get an event rate of 1.16, and then calculate the sample size based on that. So that's a quick overview of these. I know you may not have seen this non-inferiority before, but it is more and more commonly used these days. There's guidelines for this. It's well understood. There's some still tricky statistical issues. For example, when we do a superiority study, we want to find the big effect. And if we have sloppiness, too much variability, we don't do our study well, we are biasing towards the null of no difference, of no effect. But if you're doing a non-inferiority, then no difference would be good for you. And so people say the intention to treat principle is the preferred method for superiority hypothesis, while the per protocol is the preferred method for non-inferiority hypothesis. And when we talk about these two on tomorrow, okay, we will then be more clear on this. Yep. Yes, that's FDA. So if between the 50 and 75, you cannot. So actually, the 50 is the minimum. They would, depending on what in how important it is for that particular drug, they may require you to do 75. That's why I say it's very important if you're going to submit something to the FDA, you're doing a non-inferiority, you draft your protocol with this, and you go and consult with them first before you finalize the protocol. And you say, we are doing this, we want to use this margin. What if we are able to show this with this margin, this proof? Because if they say no, you may have to change it. But it's just advice. But if you're not submitted to the FDA, you don't have to do any of this. Okay? All right. So, well, actually, we can talk about this now in who to include in your primary analysis. And we started talking about this. And ITT is simple definition. Everybody randomized is included in the analysis, no matter what. If I don't have the information, I have to make it up one way or another. Right? And people do. So that is simple. And the protocol is all randomized that followed the protocol perfect. Again, not ambiguous whatsoever. Now you will see in the literature a bunch of different terms. Not standardized. Who knows what they want to mean? People use the word modified to just say modified. And they won't even say what the modification is. And you don't know what it is because the numbers from one table to another vary. And they go back and forth. And who knows why the journal let them publish this kind of garbage. But people do this kind of stuff. So there is modified ITT where all randomized that were actually eligible. Oh, there were like 28 people that should not have been randomized. But because the lab result did not came on time, we had randomized them, and therefore we don't want to include them. Or all randomized correctly. Oh yeah, we made a mistake, and these two people should have gone here, but they picked up the wrong envelope, they opened the wrong thing. Somebody did something wrong. This people went there. They should have gone here. So sorry, let's do that. Or they were randomized, but then we actually didn't give them that intervention for some reason. Now this is even more problematic because what is that reason? Is it because the physician didn't? Oh, no, this is my friend, and I don't want to give him that one because I think this other one is better. I didn't give him that one. So this is very tricky. Or all randomized that actually started their intervention. Oh, because they actually did not even come. We randomized them. They said they were coming, but they didn't come to pick up the pills. They didn't want to do the exercise routine. They didn't want to do this. So they never showed up. So why do I include them? Again, that's also very ambiguous. Okay. Then there's people that do this. All randomized that completed enough of the intervention so as to get its effect. What is enough? What you think is enough? Because you have tortured the data sufficiently that if I analyze it this way with this people and this people, I'll get significance. If I do everything, but this is actually these, as we go down here, these are problematic. This, many times you can argue. But one thing is to say, we did modified ITT. Don't use a word without explaining what you mean. What modification did you do? Who you are not including? Who are you including? Why you're not including them? Does it make clinical sense to exclude them? Are you biasing things by that? Maybe there is a bias. I should not have had my clinic open on Saturday. And these people came on Saturday, so I don't want to do that because normally I don't have my clinic open on Saturday. That doesn't make sense. Why? Maybe some people that come on Saturday are the ones that are different than the people that don't come on Saturday because they're non-working or they're working class, and they're different. So you have to worry about these things. So the FDA is very careful on these things, and they go into all of these things. I don't work so much with the FDA, but I know a lot of people in pharma here may be wanting, your companies may be submitting things to the US FDA. So please take a close look at this. So this is just to summarize the way I like to interpret results from trial. Because let's say I've designed a study with this my delta for superiority. This is my capital delta for non-inferiority. This is my gamma for equivalence, okay? And what I really don't like is situations like this. You said when you designed your study that delta was the smallest meaningful difference to say this is a better thing. And you get a confidence interval that the upper bound doesn't even go beyond this. It's the meaningless values of difference. But because this doesn't include 0, hey, this is statistically significant. This meaningless small difference is statistically significant. So fantastic, let's celebrate it. But it's garbage. You yourself said, we think this is the smallest meaningful difference. And I got something that's really small. But none are willing to say that. They want to say significant because that magic word significant is there. You say this is a significant, unimportant result. Would you say that? You should say that. This is a statistically significant, meaningless result. Should have the guts to say that. People don't do that. We'll tell you that.
Decide a sample. We developed the software. We reviewed all the softwares. There are two situations here. I also had a discussion with Shrikant. When we decide the superiority trial sample size, we incorporate the delta. How big the difference should be to say this is superior. But when you take a testing hypothesis decision whether it is significantly different or not, we never look at the delta. Meaning, supposing my difference is 5 units, my delta, what I have used is 6 units. I will not look at the 6 units. I will say whether the 5 is different from 0 or not. It is statistically significant. The whole, that's what Kant is saying. The whole world is moving towards significantly different from 0. 0 never gets used in the sample size calculation. The only one situation we use this delta. When you compare your intervention with a placebo, so when you do the placebo arm, you wanted to see the difference is greater than the delta. So the delta is attributed to the placebo effect. Therefore, whatever you get should be much more than the delta. Right. So that is how they design.
So no, I think that's a good way of thinking about it. Is that just like the delta that is used for the non-inferiority is based on preserving the effect of a reference, one could think about this delta that I'm using here should be the placebo effect, the minimal difference that would be there.
Another thing which you said, that equivalence, there is the concept all over the world. Because when you say equivalence, we had a lot of problem with this concept. Say equivalence, there's a small difference which we are talking about. Therefore, obviously, your sample size has to be. There is a book from Southampton. I forgot the name of the book that is dedicated for sample size. It's a good name. I'll tell you the name. It's there now. It's a sample size calculation. Campbell.
Oh, Campbell.
Campbell book from Southampton. What he suggested is that when you use This is especially this area. Because you were saying coming closer to zero. When he said you talk the difference between the two arms is 3 units, 2 units. Why not 0 units? So his concept is that delta becomes 0, then you calculate the sample size. No, it will come down.
It'll come down.
Because, so I will explain that. I will show you this concept here. So we don't use that delta at all in the non-inferiority designs to calculate sample size. It's basically that you say, what is your delta? You use it that much. When you don't bring it, the delta minus the two differences into the calculation at all. So that is something. So I'll explain this tomorrow. When I put that formula and explain it, you will understand what we are talking about. Then the life becomes very easier. That is how the whole thing that is something. Everybody goes with the concept of the smaller difference. The number has to be very large. That it doesn't have to be.
Right. So then the proper interpretation would be if you had something where your confidence interval is the lower bound is above your smallest clinically meaningful one, then you're very meaningfully better. If you are predominantly above, even though you're, some of it is below the clinically smallest one, but the majority of it is above, then you can say it's really that much better. But, this issue of whether you're comparing against zero becomes the problem, because really we should be able to, or willing to say, we found statistical significance, but it's not meaningfully superior. The reason this can happen is if I do a very large study, then I get, because of the large sample size, I have smaller standard errors, and therefore the width of my confidence interval becomes smaller. I have a more precise confidence interval, but I still am not getting a big effect. So you can have small effects that are now statistically significant. I think that was it.